So today I want to talk about gene duplication, which is a very important subject in molecular evolution. And also I want to review some of historical ideas on gene duplication, how we recognize gene duplication as an important event in evolution. So to talk about gene duplication, actually the slide should have like three different phenotype of Drosophila. One of the earliest observation of gene duplication was the discovery of the bar locus in Drosophila. So showing here on this slide should be one of the wild type round eyes of Drosophila, and the other one is the heterozygous bar over plus, and that is the one showing a kidney shape of Drosophila eye. And the third one is the homozygous female, and this homozygous female have two mutations, the mutant alleles, and showing very narrowed eye shape, and that's why it gets the name bar eye. So it's a very narrowed eye shape of Drosophila compound eye. So the mutant was discovered in 1913, and the paper was published in the following year. And the discovery of this mutation was not a very surprising thing because it's a mutation on the X chromosome. But what happened in the stock was really strange to most of the researchers at that time. So the unexpected observation is in the bar stock, so in the mutant stock, the homozygous bar mutant stock, then supposedly every single fly will have the same phenotype for male and female as well, then over generations. However, occasionally people will observe wild type revertance. So when we see a wild type stock, like a wild type fly in a mutant stock, the immediate thought, okay, is this a contamination? But every once in a while, they discover the wild type fly from the stock. So this seems not a sort of like exceptional contamination case, and actually it's an exceptional observation of this mutant stock. And more than the wild type flies, they also observe a fly with very narrowed eye shape, and they call it ultra eyes. So this seems like really strange because the mutant itself seems not stable all the time as others. And by that time, they already did discover fork, yellow, white, all those X-link mutations. But only this one is really something problematic with this stock. So they start doing some research on this stock, and by setting up crosses. And using the crosses, Sturven and Morgan hypothesized. They think actually the observation was every time they observe the wild type revertant, when I say revertant, it's like it becomes from the bar mutant revert to the wild type state, or the ultra bar, like the even narrower eye phenotype. In these two extreme phenotypes, every fly will the originate of this phenotype is they are always associated with crossing over. So the crossing over is the exchange between two homologous chromosomes. Then they further estimate the recombination rate and hypothesize the recombination is occur right at or near the bar locus. So they hypothesize the unequal crossing over, and this is sort of a diagram showing in the present time textbook. Will show down the eye colors, eye shape are showing in this slide. So the first one is the wild type, with the genotype. So they use the one red color as to indicate the bar locus. Then the heterozygous bar means the bar is a duplication of the original allele. So that was the hypothesis. So because of that, in the homozygous female, those type of females contain 2 bar mutant. And actually you can see the 2 red blocks on each chromosome. And if the unequal crossing over occur between these 2 homologous chromosomes, it could result in a single copy, that's the revertant to the wild type, or a 3 copy of the bar locus. So in that case, you will see the ultra bar. So that's the explanation they hypothesized to explain why every once in a while we'll see unusual phenotype recovered from the bar stock. Okay. So there are several ways to test this hypothesis. One is to look at cytologically. So by the time they did all this experiment, it was not possible because there's no polytene chromosome. We don't know polytene chromosome until 1930s. So there's no way to examine this idea at the cytological level. But they also did crosses using like a recombination, using specific genetic recombination to make sure indeed the ultra bar and the revertant wild type was discovered or was generated by crossing over at the bar locus. So the observation cytological evidence for this duplication was done by Bridges. So Bridges did the beautiful cytological observation on the polytene chromosome of Drosophila. So as shown in this original figure from his paper in Science, and you can see at the cytological locus, he can illustrate the bar indeed has a duplicate sequence of the wild type. Then after the unequal crossing over, you will regenerate the wild type locus and also the ultra bar. That's the 3 copy of the bar sequences. So this is like before we have any DNA data or before we know DNA is the genetic material. They use both genetics and cytological evidence to demonstrate bar mutant is a duplication on the X chromosome and result in this mutant phenotype. Okay, what is unequal crossing over? So some of you took genetics before, probably already heard about it. So an unequal crossing over is illustrated in this slide. And this slide showing the duplication event occurred between 2 homologous chromosomes. Showing on your left is the slide we have A, B, C, 3 different genes. And the recombination usually occurs at a homologous region. So in our genetics book, we usually talk about crossing over or recombination occur, for example, at the A locus or between A and B, so we can calculate the recombination rate. But here, unequal crossing over actually exchange the 2 DNA strands at a different site. So if you look at this slide, it indicates the 2 red dots, and that's the exchanging site. So you can see they are not aligned together, so they're sort of like at a different location based on their homology. So if that is the case, crossing over occur between the 2 black spots. Then they will result a duplication of the B locus showing here. So in this case, you can see the duplication segments and contain the entire B region, so that B got duplicated. So that's the hypothesis how the bar mutation was originally generated. It starts from a wild-type chromosome, and unequal crossing over produce a duplicate copy of the bar. Then this can be expanded at the later stage, as shown in the right-hand side of the slide. So now you have two B locus, and the B is homologous with its paralogous. So now you have two B's. You can label B1 and B2. And they are very similar because they originate by gene duplication. Then any position among the two homologous chromosomes could happen in recombination. So if that is the case, you will have like 3 copies of the B locus. And this is how unequal crossing over generate duplication segments in our genome, in Drosophila, but also in other animal or plants.